Sure. So in this case, along the rows, you're looking at a specific gene. So each one of these squares represents a spot on the microarray. This is for the gene ETS2, and each column represents a different cell sample. And whether or not, in this case, whether or not that cell sample was expressing or had a low level of H1P, a medium level H1P, or a high level of H1P. So that's what this little wedge represents. And so what this is basically saying is that, at least for ETS2, when you take a look at H1P expression, once you get moderate levels of H1P expression, you start to see ETS2 expression. Did I answer your question? Yeah. Any other questions? Yeah.
So the red means that there's no expression?
I see. So in this case, instead of looking at two different cell types, you're looking at one cell type. You're looking at either upregulation, which is green, or downregulation. So if this is your baseline where you have, in this case, no SOX2, once you start adding SOX2, the expression of these genes drops, which is why it's rare. In the case of TCL1, whatever your baseline expression level is, once you start adding TCI1, you start seeing an increase in expression, which is why it's green. Any other questions? Okay. And so the whole idea is that you want to come up with some sort of understanding of whether or not you're seeing an increase or a decrease in expression. And the way that you do that is by just taking the log of the intensity of the red divided by the intensity of the green. That gives you kind of this value that you can use to say whether or not it is upregulated or downregulated. So again, you're really just looking at the ratio of red to green of any one particular spot. Any questions? So again, the whole idea is that you are collecting RNA from different cell samples. You're hybridizing that RNA to a chip with the genes on it. And then you're examining the relative levels of RNA associated with any particular gene. So therefore you get an idea of which genes are upregulated, which genes are downregulated in a specific cell type or in a cell under different conditions. Any questions about that? All right, well then a second way that you can take a look at overall gene expression is through a process called RNA-seq, or RNA sequencing. And in essence, it is just a more modernized way of doing the same thing that microarrays do. And so again, you can ask pretty much the same types of questions that you can with a microarray, except for the process is slightly different. All right, so then with RNA-seq, again, what you do is you isolate RNA, and then what you end up doing is you take that RNA and you break it up into pieces, and then you make a cDNA. Once you get the cDNA, what you do is you kind of tack on these ends to it that basically allow you to sequence off of the RNAs, string off the cDNA, so that then you actually, instead of looking at hybridization like you do with a microarray, you're actually doing direct sequencing of the cDNAs. And so you're actually directly sequencing the product from the mRNA molecules. So it actually gives you a, instead of having to go and take a look back at the chip to determine which gene is actually being hybridized to, it actually gives you DNA sequence readout, so you know exactly what gene is being expressed. And so again, you can show this in terms of green being upregulated and red being downregulated. So the output of information, the way that you analyze the information, is kind of the same as you do with gene chips. Again, though, the difference is that instead of depending upon hybridization, you're depending upon DNA sequencing. Any questions about that? But again, you can ask the same types of questions. Yes?
I have a question about the previous slide. How specific are those RG1 and RG?
How what?
How specific are those adapters that
Mhm.
you attach to the DNA?
Right.
What
So you attach these so that you can do two things. One, so that they act as primers for sequencing.
and those are different primers for different DNA keys, or they're just the same?
They're just the same. And then two, they do this little barcode thing here, which basically allows you to tell what samples these came from, whether or not they came from nerve cell or liver cell or muscle cell or cells treated with glucose or not treated with glucose. It's just kind of an internal identifier of the original cell. Any questions about that? So then, given the fact that RNA sequencing and microarrays kind of give you the same information, and you ask them questions, why do one over the other? Well, RNA sequencing has some advantages. First and foremost, you don't need to make a gene chip. So you don't actually need to make a reference sequence for the genome. So what that means is that, for example, if you wanted to take a look at the transcription profile of an organism whose genome had not been sequenced, you could not use microarrays because since you don't know the gene sequences, you cannot make a gene chip. So gene chips can only be made for those organisms that we have DNA sequence for. Therefore, you're limited in the organisms that you can examine. Since RNA sequencing doesn't really depend upon knowing what the genome is, since it's all based upon actual sequencing of cDNA molecules, you don't need to know thing one about the genome that you're sequencing. That makes sense? Any questions about that? All right. Second, it has low background noise. So the signals that you get as far as 3-fold, 5-fold, 10-fold more transcription, less transcription is much better. You have a higher dynamic range. Meaning that you can see bigger differences in transcription. And most importantly, you've got high reproducibility. One of the problems with microarrays is that oftentimes you have to do the same experiment 9, 10 times an average across that, because you can actually see widely different results depending on how well the hybridization worked. The major disadvantage of RNA-seq has to do with the fact that it's fairly expensive. However, as sequencing becomes cheaper and cheaper, this is becoming more feasible. Any questions about that? All right. So then we've talked then about how it is that we can take a look at two different cell types and determine what their transcriptional profile is to get an idea of why the nerve cell is a nerve cell, why the muscle cell is a muscle cell. But we still really haven't talked about how it is that those differences get established. So how is it that you establish differences during development? And so how do you get that initial difference? You've got that one fertilized egg cell. Eventually you get these multiple different types of cells. How do you get that initial differentiation? And then once you get that initial differentiation, how do you amplify that so that you get multiple different cell types? Well, that first step, making that initial differentiation, that process is called symmetry breaking. In other words, going from the same cell to two different cells. And there are basically two ways you can go about doing that. One way is through asymmetric cell division. In other words, when that cell divides, there's something different between the two daughter cells. The other way is having cells in different environments. In other words, the two daughter cells are identical, but they're exposed to different environments. And that is shown in the next couple of slides. So again, this whole idea of breaking symmetry, somehow going from one cell to two different cells. So again, you can have asymmetric cell division, where the idea is that for some reason, there's